Great. So, with the wonders of technology, we finally get there. As I've said, I am lead the Free Radical Biochemistry group at the University of Canterbury and have an honorary position at the University of Otago School of Medicine here in Christchurch. I'm also the Principal Biochemist for MARS Bioimaging, which is a company that has built and designed a series of X-ray machines that we are designing to actually be able to see the arteries in people's necks that lead to stroke. So the talk today I'm going to show you is how we are tying this technology together. So as a disclosure, I must say that I am partly paid by the MARS Bioimaging Ltd group, and I do have shares in the company. Small shares, but still do have a shareholding. I'm also a member of the CMS group, which runs the Large Hadron Collider in CERN in Geneva, which is quite cool, I suppose. We get to go to Geneva and go down into the tunnels. So we'll just go to our next slide. So we work on cardiovascular disease. We're working on cardiovascular disease for a long time. And this work started when I finished my postdoc. Well, we actually finished my PhD at the University of Otago a long time ago. And there I studied sheep lipase, and then I ended up at the Heart Research Institute in Sydney, where I did a lot of background work on proteins and their oxidation, and then got into cardiovascular disease. And my next postdoc, I went to Austria, keeping with this theme of countries beginning with A, and there we studied the whole process of the cholesterol particles in your blood getting damaged. I then came back to New Zealand and set up my own research group, and over time we've gone from looking at this fundamental process of the damage through to how cells respond to actually being able to see these cells doing things actually within the body. So what is cardiovascular disease? It's one of the big causes of stroke. I'll just move my camera down. We're seeing out of my eye. You're seeing sort of the top of my head. So cardiovascular disease is often portrayed by some doctors and also by the news media as a buildup of cholesterol, fat in the artery wall. This is actually completely and utterly wrong. What is actually building up is pus, white blood cells in your artery wall. Now, those white blood cells are full of cholesterol and fat. So they're very fat pus, basically. And if they do rupture, they will deposit material, but it's all inside the wall. It's not on the surface of the artery. It's actually inside the wall. So I'm going to show you a drawing of that in a minute. Now, if this sort of thing, this plaque in this little drawing here ruptures, this pus breaks out. It doesn't ooze out onto your skin like some sort of infection on your hand. Of course, it's rupturing out into the artery, into where the flow of blood is, and the body instantly responds by forming a scab, a clot. Now that clot can go off, and if it's in the heart, it's going to block the blood supply to parts of your heart. You're going to have a heart attack. In the case of a stroke, the carotid artery, which is this bifurcation here in your neck, if you actually put a hand under your jaw and press hard, you can actually feel that bifurcation pulsating away. But you get a lot of this build-up around there of this pus, and with time that can rupture, and the resulting clots can go up into the brain and, of course, cause a stroke by blocking the blood supply to part of the brain. So we've been studying this process in quite a lot of depth. So when you're born, you've got nice pristine arteries, and an artery consists of this tube made up of muscle cells. And that's what makes it really elastic and really responsive. On the inside of that layer is this basement membrane, which is sort of like a waterproof coating, but also forms a sticky part between the muscles, and the final layer, which is the waterproof layer, in a sense, the endothelium cells. So these are cells lining the inside of all your arteries. There's a continuous mosaic of these sort of like tiles going through. Now, what happens is, we don't know exactly what's driving it, but inflammation occurs in here. And this is made worse by the presence of higher levels of cholesterol and lots of other factors, such that you end up with white blood cells coming into the artery walls and causing the wall to balloon out. So this is a pussy growth inside, not on the surface, but inside the artery. And of course, this bursts open, it can get very serious. So one of the big problems is that growth is asymptomatic. So the line we use is cardiovascular disease is asymptomatic. In other words, it has no symptoms. You don't feel anything, until something happens. Unfortunately, for a large number of people, that's death. A heart attack, a stroke, and as you know, death is always not the worst thing that can happen. You can end up somebody very disabilitated, all sorts of problems as a result of that blockage of blood supply, particularly to the brain. You also have lots of other presentations. People can lose sight on one eye. Also ischemic attacks where they just have a small mini stroke where they pass out or lose consciousness for a few seconds. So generally, until those things are happening, there's no other symptoms. So it's a quiet, nasty disease. The big problem is nobody presents with a thickened artery. And this is the fundamental problem. You can't actually see these things because they're actually quite small. The thickness of your average plaque that causes a stroke is only 2 to 3 millimeters thick, and that barely shows up on imaging. So this means that people are identified as at risk. And that means people with a family history, smokers, diabetes, people present with high blood pressure, which is a consequence. Blood cholesterol, not the greatest marker, but it is something. And the higher the cholesterol level, the higher the chance of having a stroke or a cardiac event. So these things are related. Age is actually the biggest risk factor, followed by family history, followed by smoking, and then diabetes. Unfortunately, as a result, we're limited in how we measure treatment. So treatment is usually based on lowering the lipid level and reducing those secondary symptoms like high blood pressure and other sort of symptoms. So it's a real problem. So we can't really, with the current systems, be able to see the plaque size, work out whether the plaque is stable and might rupture, and more importantly, we can't see the inflammation event that's leading to this pus buildup in the first place. And it's only recent over the last 5 or 6 years that we've actually been really seriously starting to look at measuring the inflammation. One of the problems though is knowing whose inflammation to look at, because you can measure the inflammatory markers, but you're not quite sure whether they have cardiovascular disease or not until the disease is very advanced. And like cancer or any other disease, you want to get it early on. So the markers we have are not great. So just to get an idea of the actual underlying mechanism. So you've got blood flowing through here through the white part, and this is a cross section of an artery. This is actually time going along here. You see my cursor going along here. So we're starting off with nice pristine endothelial cells, and some sort of damage happens in the endothelial lining. We don't know what that damage is. There's lots of theories, lots of arguments, hasn't led to fisticuffs at conferences, but it can get quite passionate and heated. It seems that cholesterol makes that process worse, but there's a lot of different possible mechanisms for that. The end result anyway is you end up with these monocytes, which is a type of circulating white blood cell, burrowing into this lining like they do with any form of inflammation, and they turn into these quite large macrophages. They actually grow in size fourfold, and they start picking up cholesterol, and they start picking up both damaged and aggregated cholesterol particles. So the cholesterol particles are called LDL. They're the ones that apply cholesterol to your body, but they somehow get damaged, most likely by oxidation within this wall of the artery. And the macrophages not only are producing free radicals that can damage the oxidized LDL that seems to be stuck there, they also take it up. And if they take up more than they can process and get rid of, and this is where high cholesterol seems to make it worse, you end up with the macrophages becoming foam cells. So these are big fat cells. They're like, if you take me, give me really good television to watch and a lot of cream donuts, I'm going to end up stuck on the couch, not going anywhere. And this is what they seem to do. They remain stuck in here. And this funny looking cell here is actually a T cell. They're the controlling cells, and they seem to be telling them to stay put as well. With time, this growth gets quite large, and these cells are spilling out inflammatory markers. Remember, these are white blood cells. So this white blood cells are full of foam, full of cholesterol, and they start to attract other cells, including the smooth muscle cells. So you end up with a tissue that's quite complex with muscle cells, collagen laden over here, and this builds up so large, eventually it starts to pop and get ruptured. You get that thrombus formation, which is the fancy name for a blood clot. If that blood clot breaks off and goes downstream, it can block a small capillary or something, so you end up with a stroke. So you end up with the tissue dying because this thing's blocking the blood supply. Just this growth here becomes a problem because it restricts the flow of blood up into the brain when it's occurring in the carotid artery. So we've been studying the inflammation system of these white blood cells, they're called macrophages, in a number of different settings. And what we've been centering on is this compound called dihydroneopterin. So those controlling T cells, so go back, these ones down here that basically shepherd the macrophages and tell them what to do, produce a chemical called interferon. This is the one that makes you feel quite sick when you get the flu. But low-grade levels being produced in the plaque causes these macrophages to start producing this chemical called dihydroneopterin. So very recently, going the wrong way, we've just found that this dihydroneopterin actually scavenges superoxide in chlorine bleach. These are two major oxidants produced in the body to kill cells such as cancer cells, virus-infected cells, COVID-infected cells, and bacteria and fungus. So the body produces these deliberately by the macrophages and another type of cell called neutrophils to kill things off. The macrophages make this chemical to neutralize these things so the macrophage doesn't get killed by its own weapons, its own superoxide and hypochlorite. There's this thing is the reaction that neutralizes it produces this chemical called neopterin. Now, neopterin is super fluorescent. It would be a great pigment to use and stuff if it was in fact that fluoresces still in the sort of infrared, in the UV range. So you can't actually see its fluorescence by eye, but we can pick it up with machines. And it's very, very potent. So we can measure tiny concentrations of this chemical in the blood and urine of patients. We've done this in stroke patients, heart disease patients, septicemia patients, did a huge amount of rugby players using it to show that, yes, when a winger runs into a forward, there is serious inflammation. We got big spikes from that. So we did a lot of work with the Crusaders and the Canterbury rugby team, showing this whole system is a really good marker of inflammation. So we measure it using our HPLCs. These are two of our HPLCs in the laboratory. This is one of my students who actually developed the current form of the assay. He's actually hamming it up. We don't use spanners that big in this machine, but we put our samples in here. The samples are sampled, the liquid's taken out of the urine or plasma, pumped around through this machine, and the fluorescence is checked in this machine here. We've got another one in a separate unit over here. And what this looks like on our printouts is a great big spike of fluorescence. So you can see here on this chromatogram, as we call them. So this is the fluorescence, and out comes our chemical, big spike, always comes at 4.9. So we can actually detect this neopterin at very low concentrations. And if we see this elevated in the person, we know inflammation has occurred or is ongoing. So we've been getting live plaques from patients. So people were presented with stroke. One of the treatments is to open up the carotid artery. I'm going to show an image of that very soon. And the surgeons will remove this to restore a good flow of blood and remove this tissue so it doesn't rupture and cause any further strokes. So we get a call, we pop in to the hospital within half an hour of being removed from the patient. It's put on ice straight away. We grab it, bring it back to the lab, slice it up very thinly, and then put it into a dish, keep these segments alive. We can keep them alive for up to 5 days. After about 5 days, they start to fall apart because we don't have the blood flowing and all the pressure in there. But what we can do is if we add the interferon, we can actually see the levels of neopterin going up. So along here on this graph, 1, 2, 3, 4, each one of these is a separate slice of the plaque that's been cut up, starting from the front going through to the end. For a matter of scale, this is only about 1.5 centimeters in length. This is actually a very large one. You can see this thickening is very thin. Now this is a very advanced one, but this normally, look in number 2 here, this is only 0.5 millimeter or less in thickness, and it's just expanded ever so slightly. So it's not a big change, but when you cut it open, you can quite easily see it, but it's very hard to see in the imaging. But you can see that we can take these plaques chop them up and then grow them to the dish and actually look at the inflammatory state and actually get quite a lot of idea what's going on within the inflammation system. So one thing we have been doing is also getting the blood from the patients following cardiovascular disease, or following a stroke, I should say. And we can see that when we take their blood and put it through a machine and look at those who've had a stroke, so they've had their carotid endarterectomy, they've had the tissue removed, so we know definitely they've got cardiovascular disease because there's the plaque in the dish, so it's 100% definite. We compare them to people of the same age with no symptoms, we can see the level of neopterin is higher. And also if we look at total neopterin, so this is all the neopterin, both the oxidized and the oxidized form, so it's a direct measure of how activated that white blood cell is. Also that's elevated when you compare the control to the total neopterin. Now, when we generated this graph, we first made it as a bar graph. So these went down the bottom. We thought, oh, great, these graphs, one's bigger than the other. And then we put it into this form because we were going to go to a medical conference and presented it. And then for the next 24 hours, we basically did a major panic and stressed, because if you look at these graphs, so this is where majority of the numbers for these people are occurring. And you can see there's an overlap with the control people and those with a stroke. So this person here who's at the very top, they had a huge amount of neopterin, but no symptoms compared to a person who did have stroke. So what's going on? Why is this overlap? So we're a bit concerned about that. And then we suddenly realized that the problem was, how do we know our controls are controls? And this is the fundamental problem with all research on heart diseases. Until it gets advanced, remember my very early slide that we only detect it when it's advanced. How do you know your controls are controls? So this becomes a huge problem for any studies. And this has hampered cardiovascular disease for a long time. We can't look into somebody's artery with the current imaging systems and say, you've got pristine arteries, you haven't. You've got cardiovascular disease, and look, these are the markers that go up. And they're going up, but in this person, they're not, even though you're of the same age. So it's very hard to separate it out. So this is what we're trying to achieve very ambitiously in our research. So the fundamental question, are our controls really controls? Do they have no cardiovascular disease? So I'm going to switch from talking about biochemical stuff to the area of imaging, which is an area we've been involved with, starting off very small and then becoming more and more such that it dominates half of our research now in my laboratory. So this is a CT image of a person's artery. This is actually of the artery going past the hip. I'm using this one because it's actually a really nice clear image compared to some of the other ones I got. And you can see the artery is a bit fairy in that we've got calcium deposits here. So you can see bits of calcium, but we can't actually see the lipids or the growth. There's probably a whole growth in here that we can't actually see. And this is how the problem with X-ray machines. You've got, if you look at the bottom here, you've got X-ray source blasting out X-rays. You've got an object, i.e. a person sitting there. And what we're looking is the shadows. This is the X-rays being absorbed. It's literally a black and white shadow box. We're just looking at the shadows. We're not getting any information about the type of material that the X-rays pass through. So all we can say is this bit absorbs a lot of X-rays, this bit doesn't. But what is it? It's a real problem. This is highlighted even further if we take this pile of test tubes in this little holder being stuck into one of our machines to be imaged. And you can see in the classic CT image, there's a shadow box. This particular tube absorbed a huge amount of X-rays, but all the other ones are very similar. What are they? They're all, actually there's a real mixture of different chemicals here, but you can't tell because all you're doing is counting the number of X-rays get through. Now, the thing is, X-rays, going to this next slide, actually come in a range of wavelengths or energies. So I'll just stick to talking about them as energies. In a sense, you could say it's like colors. So with visible light, the red light is low energy, and the more intense light, with blue light, has more energy in it. And of course, you get into UV light, which you can't see, has got even more energy, hence it causing burns. You've got the same with X-rays, different energies of the X-rays. And different chemicals absorb the X-rays in different spots. So calcium absorbs X-rays really well around 40 keV, whereas when you look at water, it doesn't absorb all that well, and it's quite even all the way through. Lipids tend to absorb less than water. So if we take this image, put through X-rays through, well, take this simpler tubes, put the X-ray through, and use what's called a Medipix detector. It can detect the different energy levels of the individual light rays, individual photon particles of light that go through at any stage through the object. And from that, you can work out with the CT machine what is the spectrum for each different thing, and then get your computer to identify that and say, oh, that tube is lipid, so there's that tube there. Can't really tell much difference between those two, but that is actually lipid and that's water. So it's all lit up. So we've now got the spectrum of the X-rays telling us what the different chemicals are. And this then allows us to distinguish different types of tissue, not just say that's calcium, that's something else over here. So if you take one of those carotid plaques that's been cut out of a person who's had a stroke, and then we put it through the imaging system with our Medipix detector, we can now actually see that around these two regions, so this is the same plaque, but we've digitally imaged it and then sliced it open. We can say that bit is full of calcium. There's another big calcium piece that's up in here. This bit here, this is absolutely full of lipids. Basically, it's just full of pus. So macrophage is full of pus, and this ruptured. This would have sent clots up into the brain and up into the face, basically, and into the eyes. And this person would probably have lost their sight if this ruptured. So when our surgeons saw this, they were very excited because they go on very little information with the CT machine, but to see this told them what they did with the operation was really important, and this would have saved that patient's life. So these are the actual machines. The technology is, I'll show you the next slide, is developed in Switzerland and CERN, and here in the University of Canterbury in Christchurch, as part of a joint venture company called MARS Bioimaging, we've been building these detector machines. There's a pile of them in a row here, and they're basically a specimen CT, these ones. So we have our X-ray source here, that's an X-ray tube inside that blue box. Here we have our camera over here with that spectrum color imaging X-ray camera, and it's full of these little sensor chips. So each one of those is a little chip, each one counts photons one at a time and works out the energy. So that chip there is a 128 by 128 array. So it's like your electrical camera, works in the same way in some ways, but the key thing is it can detect X-rays. So you put your specimen here, and this whole thing rotates around as a gantry. So it does the classic CT circulating around to give you the 3D image. And you can see just the top of the gantry inside one of these machines. These are our ones that we've built for laboratory experiments. And here is our commercial machine now. We're selling these. And this is actually part of a clinical trial at Pacific Radiology. People put their arm inside there so we can image their arms and fingers and stuff to look at damage that they've had from an accident. There's Anthony Butler, who's the lead physicist and 2IC of the company. And we've also got a specimen scanner with a different frontage where you can put plaque samples, mice, other things inside here and image these as well. So you can see they're all quite mobile and fully contained units. So they said the technology came from CERN. This is down in the tunnels at CERN where they discovered the Higgs boson. It's a large particle accelerator, and they're slammed together in this tube here, and they detected the Higgs boson in this big machine here. Inside here is the prototype of these chips here. So these were invented 20 years ago, 25 years ago, and they're miniaturized and made into these systems here now, which we then have in these machines here. So what have we been doing to image plaque? We have been, our surgeons, somebody has had a stroke. Imaging suggests that the cause of the stroke is in the carotid artery. So the surgeon goes in, opens up the neck, exposes the carotid artery. There's the bifurcation there, slices open, and literally just pulls out this plaque tissue. They then sew it up and then restore the blood supply, and the patient literally walks out quite comfortable, except for a couple of stitches in the neck. We then get the plaque and we slice it up into little pieces, and we image it both before and after with the MARS scanner before we slice it up, and then we do lots of experiments looking at the inflammation process going on inside this tissue, and then relate that to the actual image we get of the plaque. So here's some very high resolution images we've generated of some plaque we've studied. This is the photograph image. This is one side and this is the other side of the same plaque. This big slice here is where the surgeon was heavy-handed with his scalpel and cut it open. We tell them to just get the plaque out and we'll take whatever we get. Sometimes they come out very whole, sometimes they've got a big slice down the middle like that. I mean, it's the patient that's important. When we image that, you can see we end up with this amazing image. This is a 3D image where we're just looking at the surface from the view. We can actually set it up so we can see classic slices and subtract pieces away. But just for this talk, we thought I'd show this quite stunning image of the plaque. And you can see there's big lumps of calcium in here, and this is very lipid, cholesterol-dense material where there'll be a lot of macrophages deposited. This red stuff is just the connective tissues, that's thickened artery wall. Because it's digital, can subtract the back of this and the front of this. You can actually peer inside here. And what we actually find is a rich iron deposit. This is discovered by one of my master's students and worked out how to do this. And this is where blood has got inside this material, and the blood has got trapped. The red blood cells have lysed open and deposited iron in here. And then this iron is cytotoxic, starts to kill cells and set off an inflammatory process that makes this even worse. So not only did this patient have a large blob here sitting here waiting to rupture, he also had iron being deposited into the wall of the artery, which could then cause an even bigger growth of the artery. While we're looking at this, and we can actually view this with 3D glasses, we noted this cave in here, this sort of gap. So we then looked at a slice. We can actually see there's an eruption here. So we go through a series of slices going down sideways. So this is peering down from the top. It's a series of classic CT slices. You can see there's actually a proper hole in here. So this black bit is this bit in here. And we suspect this is the bit that ruptured, that opened up, that caused a small stroke, perhaps a transient ischemic attack, I think, for this particular patient, that caused them to be referred to the hospital for surgery. So this little hole saved their life, because there's other ones that ruptured who would have been a lot more serious. So that's looking at the tissue while after it's been taken out of a patient. We can do a lot of interesting things there. But the ideal thing is we want to see this earlier on. So we've been working forward with much larger scanners that can actually image the neck and leg. And because there's some problems just working up to the neck, our original work we did last year was looking at legs. So peripheral vascular disease, the same disease that causes a stroke, also happens in the peripheral, in the leg arteries and stuff. And you end up with the same growth occurring here in the leg. It does tend to be larger because you don't have the same pressures of muscle around the neck or around the heart. So they tend to be more blobby, for want of a better word. But they're definitely a major threat to a person's health. So we have a prototype whole body scanner, as we call it. So this is a stripped down CT machine in a sense. So it's a rotating barrel which we can bolt various bits and pieces on. So this is how these things start off. It's basically a very simplified machine which has all the fundamental parts of a fancy CT machine you see in hospital, but because it's all open structure, we can stick on different size cameras, different size X-ray sources and stuff. And then we use a test subject like the CEO of the company, who's sporting his fancy CERN jacket here, and we can stick his leg in and image it. And we've done the reverse, of course, where we've got a patient and stuck their head in and imaged their neck as well. So I'm just going to show some of the best data and discuss how we're building up to actually now seeing the actual carotid arteries in a person's neck. So this is of a person's leg that was referred to us. 70-year-old male. They have arteries that have cardiovascular disease in them, heavily calcified. So you can see there's lots of lumps of calcium in here in the CT image. Now, the reason the arteries are lighting up in the CT image is iodine has been injected. So the iodine lights up the artery because it absorbs X-rays. One of the big problems with this technique is you can't actually see whether it's calcium or iodine, but you can see it's not a nice, smooth flow for that person's artery. So here's the actual patient's leg in a leg holder sitting in the machine. We're going to be imaging this part, the knee part here of the patient as the drum goes round. So we have to put it into a holder for this particular stage. Last year, scans were taking 20 minutes. We've actually got the time down enormously now by sorting out our computer algorithms and getting a faster way of getting information off the sensor chip into the computer. So we can actually scan a lot faster now. And there's the image. So what you guys are looking at, probably the first, I'm not sure if we've shown this to the general public yet, but you guys are actually seeing for the first time, a CT image of an artery without the use of contrast agents, so native X-rays. And we're able to see that because we're looking at the different wavelengths of the X-ray, and based on the balance, the color of the X-rays coming through, we can identify in comparison to our standard, what we call the phantoms, we can know that this is definitely calcium. These are calcium deposits in here. And here you can see the artery coming down. This is the wall of the artery, and it bifurcates here, coming down here. So here's the bifurcation point. This is actually in behind the knee, so that's where it is there. So we can see lipid-rich material that's in the leg. So various layers of adipose tissue, fat tissue in here. We've got various muscle layers here, and we can clearly see the artery. The reason it seems to disappear here is actually this is a slice. So this is in the plane of the slice. And as you come down here, it's actually moved out of the plane. It's moved below out of our image. So if we went to the next slice of our computer stack of images, we'd see the artery continuing on. So this gives us really clear imaging of what's going on at very high resolution. So this is images we're working on now to actually make them even clearer and give the surgeons even clearer information. So this is a very early scan of a carotid. So this is the carotid in here. You can see the calcification in the patient. So as I say carotid, this is in the neck artery that causes stroke. And this is the actual MARS image. We can actually see carotid artery here in the bifurcation. So this was just before we got stuck with the whole lockdown problems last year. There's been real problems trying to get supply of equipment, but that's all solved now. We're about to start really hammering away now with imaging our carotid patient, our stroke patients. And the real key thing is this is not blurred. So there was a big debate within our team, does the carotid artery pulsate? So if you take your hand, stick it under your artery, press down, you can feel it pulsating away. My argument, being the biochemist, was it's pulsating because you've got your finger on it. The medic's view was it's pulsating because it's near the heart. Turns out I was right. It's pulsating because you've got your finger on it. So we've got really quite clear images of this. And this was a very preliminary image. What we're getting now that hasn't been processed, so it's not ready to be released to the public, is a lot clearer. So we're really pleased with what we're getting out of the scanning now with our prototype machine. Later on the year, we're going to start designing brand new machines for commercial application for imaging the arteries in the neck, the carotid artery, based on the data we're getting off the prototype machine. So here we have what we're trying to achieve is be able to look at these carotid plaques in patients in situ, both people that are presenting with a stroke and people that are thought to be at risk of stroke but who haven't had a stroke. So in a sense, our controls. So we want to be able to detect the plaque at this very high resolution. This is actually a slice view of one. That red stuff is actually hemoglobin down there, which is quite an interesting plaque, that one. So what we want to be able to detect the plaque in patients. We want to be able to say the state of the plaque. Is it going to rupture? Is Is it stable? Should the surgeon operate? It's a risky operation. Anytime you put under anaesthetic and have your arteries opened up with somebody with a knife, that's a risk. So you want to know, is the statin therapy or other drug therapies, are they working? And if they're not working, then you want to say, well, we should send that person to surgery. But is the plaque unstable? Yes, it is. We'll send them to surgery. So this is the sort of questions we want them to develop. More importantly, we want to, well, the surgeons say that's really important, but I want to actually go one step further. And this is what a big chunk of our research for the Neurological Foundation is, to take our stroke patients and be able to identify markers that are specific. These are chemicals released from this inflammation process in the artery that is specific to the growth of that plaque. So we can take a blood sample or a urine sample, more likely a blood sample, from a 50-year-old person that we suspect, because of family history, might be developing something like this, and we can test it and go, actually, these things are elevated, we should send you to imaging, or these things are low, you're fine. We'll have a look in a couple of years' time. So we want to be able to develop a blood test or a urine test or a mixture of bodily fluid tests of the inflammation markers, which we then can confirm with this spectral imaging. The other big thing is, one of the big holdups for any development of drugs that prevent stroke is that it takes time. This is a disease that probably starts when you're in your early teens and doesn't progress to being something serious to late 50s, 60s, 70s. So it takes a long time to develop. The problem with drug treatment is, therefore, you have to give the drug and then wait 10 years or 5 years to get statistical data. And that's because it takes a long time for it to get bad enough to actually pick up what's going on. With Spectral CT, we'll be able to peer inside the arteries and be able to say, this is what's going on. And also with the Spectral CT, we'll be able to confirm the biomarkers of interest and be able to go, actually, your drug treatment has reduced this down, the inflammation has stopped, and therefore you're stable, and we can look at the artery and also confirm it's stable. So it gives us a full range of options to manage people and prevent them having a serious stroke. So that is our overall goal over the next 3+ years, is to develop this technology all the way by combining imaging with biochemistry. So it just remains at the end to thank very much the Neurological Foundation for funding our research of combining imaging with biochemistry and inflammation. The machines were designed and built with funding for the government's MBIE Endeavour Fund. The University of Canterbury has been very generous in supporting the company and the various PhD students who worked on the project. Lottery Health has provided funding for my research group's study on various aspects of neopterin, and we also got funding for the Heart Foundation a couple of years ago to look at the various markers. We are indebted to the New Zealand Blood Service for giving us blood for a lot of our experiments, especially where we purify the white blood cells themselves and look at their inflammation markers. So we did a lot of work where we look at blood cells, and then we look at plaque, and then we look at whole patients. So as you imagine, you go up more and more complicated. So we often want to work back down. We've seen this with the patient. Is this happening down at the cell level and vice versa? So the New Zealand Blood Service is giving blood for those sort of studies. The vascular surgery department, particularly Professor Justin Roke, our vascular surgeon, provides us with the plaque tissue from our stroke patients to actually study the real thing. So we're quite unique in the world of actually being a research group that actually look at the actual tissue that causes stroke, as opposed to doing whole cell work or just a lot of blood work. These are some of the research students that contributed directly to the slides I've shown you today, particularly Emily Seal and Joe Healy, who do a lot of the imaging work. Raj and Anastasia did a lot of the neopterin work I've shown, and Hannah's done a lot of the plaque inflammation work that I've also shown. So that finishes an overview of where we've sort of been and where we're going.